All right, so today we're going to talk about the process called, the type of inheritance called polygenic inheritance. And it's very important to talk about polygenic inheritance because it's actually how most of inheritance works. We like to think that genetics works a lot like this, where you have a parent one way, a parent the other way, and you result in a simple inherited trait that is a result of those two parents. But inheritance is actually a lot more complicated than that, and we're going to illustrate that in terms of polygenics. And we've actually talked about this a little bit together. Pause the video here and have a go at 3 quick questions, and we'll have a look at them together in class. Last little bit of genotypes and phenotypes. So we're going to talk about the qualitative, which is good because this is really hard to talk about in terms of the quantitative treatment. So the qualitative treatment of polygenic inheritance as contributing to what we call continuous variation in a population, illustrated by the determination of human skin color, which we're going to talk about as a specific example through the genes involved in melanin production, which is the protein that makes your skin darker, or by variation in height, which we already talked about. So I want you to imagine a world of only monogenic genes. So a world where everyone is either very short or very tall or very dark or well, this guy's not very light, but very dark or lighter. All right. It wouldn't be an interesting world, really, would it? So up to now, we've really focused on traits that are controlled by a single gene, so they're monogenic. But fortunately, most traits in humans are controlled by a variety of genes. They're polygenic, and a great example of this is height. So as you can see, we are the result of a very, very large group of genes that result in people of very, very different heights. Okay? So this leads to what we call continuous and discontinuous variation. Okay, so a discontinuous variation is monogenic. Okay. Monogenic. Okay, and we have very distinct categories. There's no in between. Okay, it tends to be qualitative. Okay, things like tongue rolling, your fingerprints, your eye color, even though I would actually argue that eye color can also fit into this group over there. But your ABO blood groups are certainly a very good example of a monogenic gene that actually has quite a few alleles in there, but it's still only one gene. Okay, we represent this using a bar graph, and it's a limited number of phenotypes, but in intermediaries, we're talking about in-betweens, okay? Continuous variation, we have no distinct categories, no limit on the value, and it tends to be quantitative. We quantitatively take the height, the weight, the heart rate, the length of someone's finger, the length of a leaf, okay? And we represent the bar graph, and it's often distributed in this way on a bell curve, just like we got in terms of heights of everyone at school. So there's a lot of gene and environmental influence in terms of continuous variation, and we're going to get a range of phenotypes between 2 extremes, okay? All right, so the type of variation really isn't black and white or brown and blue if you're talking about eye color. All right, the number of genes and the number of alleles within the genes are going to determine the degree that a trait is continuous or discontinuous. That's really important, that we have a degree of continuity or discontinuity, okay? No gene is fully discontinuous and no gene is fully, well, some genes are fully continuous or discontinuous, but the degree of that continuity is very important, okay? And a great example of a very continuous gene is skin color, okay? And in this example, we're going to assume that there's only 2 genes in skin color. In reality, it's about 4 polygenes and potentially more than just 4 polygenes. And not only that, but environment plays a huge role in terms of skin color, but we're going to really simplify it take the environment out of the picture, and we're going to take the extra genes. We're just going to have two. Okay. We're going to represent our alleles in a slightly different way, and it's a fine way. It's an easy way of representing it for continuous variation, and that's using pluses and minuses. Okay, so pretty much the plus allele, we're going to have increased melanin should I spot it right there? Production. That's the protein that makes your skin darker. And that's the plus. The minus allele, we're going to have decreased melanin production. Okay, so let's break this down in terms of the genes. Okay, so we still have two chromosomes that carry each one of those genes. Okay, so for example, you might have on gene 1 plus on both chromosomes. Okay, and then on gene 2 you might also have plus on both chromosomes. Okay, and that's going to result in a person who is very dark. Okay, or you could have as easily had this plus, 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 minus, all right, or plus, minus, plus, plus, all right, and then this person, they're still dark. They're not very dark. They're just dark. Okay, or you could have easily have also had plus, plus, minus, minus, or plus, minus, plus, minus, or minus, minus, plus, plus. Okay, and we're going to see this person is a nice intermediate. Okay, and then on the hindsight, we could have had plus, minus, 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 where we could have had minus, minus, plus, minus. All right, where we actually have a light colored person, all right, and then we could have had our last individual who is very light. Okay, what I want you to identify from here is the fact that we have very few possibilities at our extremes, so whether it is at 0 or at 4, and greater numbers of possibilities in the middle. All right, and so that's why this continuous variation forms for us this bell curve structure. Okay, and that's the important part of the continuous variation.